This is the lecture for eukaryotic cell parts. So we discussed prokaryotes being single-celled, simple organisms, and then we talked about eukaryotes being specialized and multicellular. So within the eukaryotic cell family, we have animal cells and plant cells. In general, animal cells are circular shaped. However, form determines function. So many specialized animal cells are not circles, but on your left-hand output page, the picture that you're going to be color coding to match your notes, the animal cell is a circle and the plant cell is the square. This organization hierarchy is just to kind of show and review that cells build tissue, tissue builds organs, organs build systems, which create an organism. But the unliving, the non-living part of this are those organelles, those parts and pieces that make up the individual cells themselves. Looking at your matrix, I need you to identify and find the row that says cell wall. It's closer to the bottom. The cell wall is found in plants only, and this is the very outside of the cell. In this picture, you can see it as the very outside line with a gap on the inside. The location is the very outside of the cell, and it's made of cellulose. That's the description. Cellulose is a sugar, and I know that because it ends in OSE. The function of the cell wall within plants is to offer support and protection. Ultimately, the cell membrane, which is just inside the cell wall, can flex. That's because plants have to take in extra materials in the event that those materials are not available. In times of a drought, if I'm not able to hold water within my cells, my plant cells will die because they won't be able to perform photosynthesis. So the job of the cell wall is to really hold in that cell membrane and keep the integrity of the plant cell while allowing it to store extra materials that it's going to need. You can pause this video and you're going to color code the row for cell wall and match it to the diagram in the plant cell, and you're going to color that cell wall that same color. Cell membranes are found in plants and in animals. Please find this row in your matrix. For animals, they are located on the very outside of the cell. In plants, they are located just inside the cell wall. If we're to go back here, you see the plant cell wall on the very outside and the membrane just inside. The cell membrane is a phospholipid bilayer with embedded proteins. So phospholipids are lipids, the word is in the word, and a bilayer has two layers, just like a bicycle has two wheels. The word embedded means inside, so those proteins are actually built inside of this phospholipid bilayer. And their function is to control what enters and exits the cell. Its job is to maintain that homeostasis. So we define homeostasis as maintaining an internal environment. Many times we refer to that as the body as a whole, the organism as a whole, but in this sense we're talking within the cell as well. We have to maintain that internal environment. So you're going to pick a color and color the row for cell membrane, and then color the cell membrane in the plant and in the animal because both have a cell membrane. The nucleus is found in plants and in animals. The location is central to the cell. That's because the nucleus's function is to store genetic information and be the control center for the cell. So having a central location makes it easily accessible to the entire cell. In plants, many times that nucleus will get bumped over if there's extra storage that needs to be had, but most of the times it is central into the cell, and it is a membrane surrounding DNA. So if you look at this picture, the blue part is labeled the nucleolus. That's the actual center, the actual core of the nucleus that contains that disorganized DNA. The chromatin surrounds that. Okay? So you have your nucleolus on the inside with the chromatin surrounding it, and all of that is your nucleus. Please pick a color for nucleus and color the nucleus in the plant and in the animal. In the animal picture, since the orientation is how it is central in that picture. In the plant cell, it's a little off to the left because you're looking down at the plant cell. So because of the different viewpoints of these two cell pictures, you just need to make sure that you're coloring the right one. Mitochondria are found in plants and in animals. They could be found anywhere throughout the cell because the mitochondria's function, their job, is to burn energy. They burn glucose during a process called cellular respiration. So that glucose, that monosaccharide, mono meaning one, saccharide meaning sugar, the building blocks of carbohydrates, get broken down by enzymes. When those big polymers, those big polysaccharides get broken down into those monomers, those monosaccharides, the mitochondria burns that for energy. So in order to make sure that I'm burning as much energy as possible, inside the mitochondria there are these folds that perform the cellular respiration. I'm increasing the surface area so I can pack a whole lot of stuff into a tiny space. So the types of cells that would have a lot of mitochondria are cells that would use a lot of energy. The example that we see many times is muscle cells or sperm cells. They're two cells that are going to require a lot of energy, specifically cardiac muscle. So your cells within your heart, that cardiac muscle, require a lot of energy because they're beating all the time, those electrical impulses. So when you see a mitochondria, it's almost like an oval with this fancy swirled design on the inside, just showing you that I'm increasing the surface area, so I'm increasing the amount of cellular respiration that can occur. Vacuoles are found in plants and in animals. Sorry, that's a typo. It says plants. That should say plants and animals. In animals, these vacuoles could be anywhere because they're temporary. Animals have to get rid of waste and take in goods. Plants, the vacuole is going to be central. That's because I need access to all of the things that I'm storing: water, carbon dioxide, hydrogen, or water, carbon dioxide, oxygen, glucose, any of the things that I need for cell respiration and for photosynthesis. When that vacuole is full, that is what can kind of offset the nucleus and push it out of the way. Because remember, we are inside the cell membrane, which is squishy, and it will allow things to move. And this is a three-dimensional object that we're looking at, a two-dimensional example. So its description, it's an empty bubble. That's what it looks like. It is there for storage only, and its function is storage. So again, in animals, because getting rid of waste is a temporary job, those vacuoles are smaller and throughout the cell. In a plant, that vacuole is a lot larger and it's really centralized. It's in a central location. And that's what can change where the nucleus is if that vacuole is full. Lysosomes are the ones that are found in animals only. Animals use lysosomes throughout their cells, and the word vesicle just means like a pocket. So it's a small pocket or container with enzymes because I need to break down waste and nutrients. So that's why the animals have lysosomes, because plants make their own nutrients. They make their own food. They're not taking in Bojangles and having to break it down into its tiny monomer pieces. They make the sugar as a monosaccharide. They make glucose. So plants need that. Plants don't need that. Animals need a lysosome because they have to be able to break down the nutrients that they take in and also get rid of the waste, because some of the things that you put into your body doesn't need all of that. And so it will go ahead and get rid of it if it needs to, using that lysosome. So again, when you're coloring this one, please only color it in the animal cell. It actually looks like another cell within the cell. So it's a circle with a membrane around it, because these are all membrane-bound organelles, meaning that they're complex parts, and it has dots in it to represent the enzymes that are breaking down the nutrients and the waste. Ribosomes are found in plants and animals, and remember that these are the non-complex organelles that are found in prokaryotic cells as well. They are located on the endoplasmic reticulum, so that's going to be that rough ER. So what makes it the rough ER is the fact that these ribosomes are actually located on the endoplasmic reticulum, and we'll see that in the next slide. So the smooth ER that we'll also talk about, which creates fats, doesn't have these ribosomes. So these are tiny organelles on top of another organelle. But they have an upper and a lower subunit, and what happens is those subunits come together and the nucleic acid in the form of RNA actually gets drugged through the opening between those two subunits. Okay, so the upper and the lower, but when you look at your paper, they just look like tiny dots. So if I were going to color this, I would color them black because they're already black on that paper. So just kind of outline this whole row in black. But their job, their function is to synthesize proteins, is to make these proteins. So you have these ribosomes making the proteins, and the rough ER, which is the rough endoplasmic reticulum, packages these proteins. So if you look at these tiny dots that are located on the ER, those are actually representing these tiny little ribosomes. So each of these little ribosomes is one of those dots on that ER. I also want you to see that the center purple circle there is representing the nucleus. So the location is connected to the nuclear membrane. So the ER is actually an extension off of the nucleus. We're seeing folds here again because I can cram more stuff into a smaller space. I'm increasing the surface area by folding in. Their function is to package and transport proteins within the cell. So ribosome makes them, and ER takes them and packages them. The second type of endoplasmic reticulum is smooth ER. And if you haven't done so already, if you create another row at the bottom of this matrix, and the term will be smooth ER, we'll fill this in. This is found in plants and in animals. And it is attached to the rough ER. So the rough ER, if you remember right here, is attached to the nuclear membrane. So I have the nucleus, the rough ER is an extension off of that, and then the smooth ER is attached to the rough ER. So it just kind of goes from nucleus, rough ER, smooth ER as it gets further away. Here I have folds, but I have no ribosomes because my job is to synthesize lipids. So the smooth ER itself actually creates those lipids, creates those fats for all kinds of things, including making more cell membrane. So because it's doing that, it doesn't need those dots, which are the ribosomes, which make the rough ER called the rough ER. In your diagram that you are color coding, there is no smooth ER represented. So you won't be coloring this row. This is just so you understand that fats are also created within the cells. So you can get fats from food, but also your cells need to combine different parts and pieces, those glycerols and those fatty acid tails, to go ahead and make sure that I have enough of these phospholipids to make a cell membrane. Ribosomes make the proteins, ER packages the proteins, and the Golgi bodies, Golgi apparatus, Golgi complex, all the same thing, ship the proteins out of the cell. So ribosomes make them, ER deals with them and packages them within the cell, and then Golgi is the one that actually ships them out. So what would I be shipping out? Enzymes. For example, that amylase. Amylase within your cells, within your cheek cells, that gets shipped into your spit to help with the digestion. Amylase starts the digestion. Pepsin works on breaking into fragments within your stomach, trypsin in the small intestine to break into smaller fragments. So Golgi is actually folded, but it's not connected to the nucleus. So yes, ER is folded, but it's directly connected. I can trace those folds back to being part of the nucleus. In the Golgi bodies, they are not. So it is folds with no dots, no ribosomes, not connected to the nucleus. So ribosomes make the proteins, ER takes the proteins, Golgi ships the proteins. Chloroplasts are found in plants only, and these are the ones that I need you to color the row and the chloroplasts themselves green. They're found towards the outside of the cell, and that's because that's where the sunlight is. So the chloroplasts are towards the outside of the plant cell, and in your diagram, they're actually represented as like, oval/triangular shapes, and they have lines in them. Those lines are representing those stacks of thylakoids. So we used that word when we talked about prokaryotes, how they had thylakoids and thylakoid membranes. And here, those thylakoids are those actual little circles, those discs that are in stacks, and that's what those lines are representing in your diagram. So these are only going to be in the plant cell towards the outside, and their job is to perform photosynthesis. You do not perform photosynthesis, you are not an autotroph, and you're an animal. So just plant cells are doing that. So I need you to color those chloroplasts only in the plant cell, that green color. Cytoplasm is found in plants and animals, and this is actually, cyto means cell, and cytoplasm is what gives my cell its 3D shape. So it's very tricky to color. I would color it white. But it's all the background in those cells, and that's because you're looking at a two-dimensional drawing of a three-dimensional process or three-dimensional object. So if you were to look at a cell, it's kind of like a real cell. It would be actually like jelly feeling because it's what holds everything in place and gives the cell its shape. So within that cytoplasm, there's also something called a cytoskeleton. Cyto meaning cell, and it's what the framework, or what we would call the bones, of the cell, and then the cytoplasm is the gel that holds everything in place. And I think about it like those old Jell-O molds that had fruit in them, where you had a Jell-O cup and then there was fruit embedded in there. The fruit is like the organelles, and the Jell-O is like the cytoplasm. So you're going to take these cell parts and color code them to match on your left-hand page. Once you've completed the notes, you've color-coded your matrix to match the cell organelles on the left-hand side, you're going to open up that cell diagram and look at that cell city analogy and try to say, if the cell were a city, what parts of the city would be parts of the cell? So reading through that cell city analogy and leaving that into your notebook to get ready. The idea here being is that you've got the information in your notes, you've looked at illustrations and color-coded them to match your notes, and now you're going to look Monday and this Friday at actual cells, and can you identify those cell organelles and those cell parts?